Yeah, we're going to be going live very, very soon. Looks like it's already set up, ready to go. Oh, it's already ready to go. Awesome. So let me get all of the things that I need, which I think I actually have already. I'm going to have to excuse my croaky voice. Hopefully it doesn't sound that different. So we're going to be going through a selection of exam questions linked to gas exchange in fish. So that is the key focus today, gas exchange in fish. And I'm just going to quickly do once over that yes, it is clear on YouTube. Yes, it's clear on Instagram and TikTok. Yes, it's clear. Awesome. Let's get started. So this is A-level biology. It is preparing for paper 1. We're going to be doing some exam questions based on this, which is gas exchange in fish. So first one we have is describe and explain how the structure of the insect gas exchange system help deliver oxygen to cells while also reducing water loss. Okay, so first thing is, it's 5 marks. I'm just going to start by just stating the different structures that there are in the insect gas exchange system. So we have spiracles connecting to the trachea, to the tracheals. And that's actually what we've got a picture of on the next page, but just to do a very quick diagram. So that'd be the spiracles. The trachea are then the larger tubes, and then the tracheals are the finer branching ones that reach lots of different respiring cells. So that would be our structures, but now we actually have to say how they help deliver oxygen, but also reduce water loss. So I'm going to stick to the water loss for now. So the spiracles, they can close, and that reduces water loss. They also have hairs around them. So spiracles have hairs, again to reduce water loss. So I've already got 3 points there. Next then, I need to talk about how the structures help with the gas exchange. So now I'm going to go back to these tracheals, because that is literally the site of gas exchange. So tracheals are highly branched. And that is large surface area. So we're up to 1, 2, 3, 4. So we need 1 more points that we can make. So the other thing I'm going to say is that the tracheals have thin walls. And the advantage of them having thin walls is a short diffusion distance. And there we go. That would be our 5 marks. Now, if you are watching along thinking, is there anything else you could have said? You could have also said that the spiracles is where diffusion happens, or you get diffusion and gas exchange happening at the tracheals. You could have also said that the tracheal walls are permeable to oxygen. You could also talk about that this exoskeleton is waterproof, so that prevents water loss as well. So that's question 1. Let's go on to the next one. So next question, the diagram below shows the insect tracheal system. That's what I was attempting to draw on the previous bit of paper. Here's one made by ChatGPT. I actually got ChatGPT to make that diagram. Pretty cool. So identify the structure through which, so first of all, we need to identify the structure through which gases exchange and leave the body of an insect. So they are going to enter and leave through the spiracle, which is A. They haven't asked us to name it. They've just said identify, but I'm still just going to put it just in case. So A, which is the spiracle. Then the tube where gas exchange happens. Now that is C, which are the tracheals. So I'm going to say C, tracheal. Part B, we then have, when at rest, gas exchange can still occur in these insects. And we have to explain the movement of oxygen into the tracheal system when it's at rest. So the reason it's emphasising this is, in the specification, it does say you need to know how insects exchange gas when they're at rest versus when they are in movement. And when they're like flying and it's movement, that's when it's to do with the movement of the water at the ends of the tracheals going into the cells because of the lactic acid being produced in anaerobic respiration. So that's that concept. But when they're at rest and they're not using oxygen up to the point of anaerobic respiration happening, the reason that it happens is just because of concentration gradients and gas exchange. So and diffusion. So I'm going to say oxygen is used up in respiration. Well, actually, I'm going to be more specific and say aerobic respiration. That is going to establish, so this establishes an oxygen concentration gradient. And lastly then, for the third mark, that means oxygen will diffuse in. And making sure you do say diffuse because it's all about the fact that we've made that concentration gradient. Right, let's have a look at the next question. Again, I got a ChatGPT to make me this diagram again so I could create these questions. The diagram below shows the insect tracheal system. Yep, same thing. But this time we have to name structures A, B, and C. Partly done this already. So A was the spiracle. B is the trachea. And then C are the tracheals. And then we have to describe the function of each of those. So the spiracles, I'm going to say, so A, where oxygen, or you could actually have gases, enter and leave. Trachea, that is connecting spiracles to tracheals, so gases can pass through. And then the tracheals are the site of gas exchange. So that's where the oxygen diffuses from the gases in these tubes into the respiring cells of the insect. Right, next question. We have to describe and explain key features of fish gills. So moving on to fish now, that make them efficient for gas exchange. For any gas exchange surface, the features we need to consider are, first of all, large surface area. Secondly, short diffusion distance. Thirdly, maintaining a concentration gradient. So all exchange surfaces have those three adaptations in common. You just then need to remember what they are for each example. So for fish gills, what provides the large surface area is the fact there are many lamellae. That'd be my first point. Next, then, I'm going to have the fact that they're very thin thin lamellae equals short diffusion pathway. Now, I didn't go for the concentration gradient, how it's maintained, because that actually links onto this question, and this isn't a structural feature, it's a mechanism that happens. So, explain how the countercurrent system in fish gills helps maximize the amount of oxygen that diffuses in the blood, and it's 3 marks. So first thing I'm going to say is what it actually means. So it's when water and blood flow in opposite directions. Then I need to say why that's actually going to be an advantage in terms of diffusing oxygen. So that means blood always passes water with a higher concentration of oxygen. And therefore, that means the diffusion gradient for oxygen, and you do have to emphasize oxygen because it said it in the question, is maintained across the entire, and that's a big thing they want to emphasize, entire lamellae. So that would be my 3 marks for that one. Right, question 3. Describe and explain 3 adaptations of the insect tracheal system for gas exchange. This is similar to the first one that I did if you've been here from the start, but this time it's not just, this one is just about gas exchange. The previous one was also about how it reduces water loss. And we have to describe and explain. So first of all, I had last time tracheals are highly branched. And that means large surface area. The next one was tracheals have thin walls. The advantage is short diffusion distance. And then we still need another one. So the next one that I'm going to go for is actually still the idea that they're highly branched, but a different advantage for this. So tracheals, highly branched, but this time I'm also going to point out that that means short diffusion distance, because it basically means because they're so highly branched, they're connecting to lots of different cells, so there's a short diffusion distance. So although I've gone for highly branched, large surface area, thin walls, short diffusion distance, highly branched, short diffusion distance, you could for this one also have said that the tracheals provide tubes filled with air, so you get fast diffusion. Or you could say that there's fluid in the end of the tracheals that moves out during exercise, so that you get faster diffusion through the air. Or you could say the body can be moved by the muscles, so that abdominal contraction and movement, and that pumping maintains the diffusion gradient as well. So you could have had that. Okay, last one for today. We've got a scientific drawing. So for this one, a student presented their observations of an insect tracheal system viewed under an optical microscope, and they produced a scientific drawing. And this was their scientific drawing. We have to suggest 4 ways the student could have improved their scientific drawing. Quick fact again, this scientific drawing was not actually produced by a student. I got ChatGPT to produce this diagram. I basically told it what I wanted a diagram of and all the things I wanted to be wrong with it so that I could create this question. So you can actually use ChatGPT to help you make more exam questions, although I suppose you have to have an idea of what the questions are like, and the mark schemes to help you with that. But yeah, brilliant. I can see some answers coming in. on the Instagram chat. It's actually the only one that I'm looking at the moment. If I click another button, I should be able to see the chat on YouTube as well. Okay, YouTube, I can see some answers coming in as well. Excellent. So we could have, first of all, they've done some shading, so there shouldn't be any shading. So don't use shading. They've also done lots of like little gaps and like overhangs, so we should have continuous lines. So I'm going to say use single continuous lines. There are more things than just that are wrong though. So we could also have that they need to add labels. So we don't have any labels or annotations. And it was under an optical microscope, but we've got no idea here of the scale or size. So I'm going to say add magnification. B. Sorry, my cat is causing absolute mischief in the plant I've got over there. So add magnification. Okay, and that's it. That was our last question, everyone. So that takes us to the end of today's live questions. Hopefully found it helpful, and I will see you next Thursday, everyone.